Speak about active matter. Let's see whether, yeah, I'll just use this. And I'll discuss about some recent work that we've been doing about the modeling of actomyosin droplets. So the two movies that are running, I will go through in a lot of detail, well, or some detail at least later on. And this is one of our simulations, and this is a crawling cell. This work has been done in the last couple of years. So most of what we speak today will be has been done in collaboration with Els and Tung, who's been a PhD student in Edinburgh and is moving to France now. Adriano Tiribocchi and Michael Cates, and Adriano and Mike are both in the audience. And so you can ask both me and them something about this work. So I'll start with a rather naive introduction on, so the problem will be trying to understand motion of cells. So trying to use active matter to understand a little bit about cell motility. So this is a crawling cell. So this is a keratocyte fragment. This is the nucleus. And this is a structure known as a lamellipodium. So this is a two-dimensional structure which is pushing the cell forward. Okay, so this is a classic example of cell motility. And if you zoom in inside that structure, if we can do that, yeah, okay. So no, I lost one. Yeah, I don't know why I lost this. So if you zoom in here, this is a network of filaments. It's like a two-dimensional networks of actin fibers. And these actin fibers, so here is the cell membrane. The actin fibers are pointing, is like a bistable distribution at the front. And they are being polymerized, and this pushes the cell forward. So this kind of motility works on a substrate. Okay? And the idea is that in order to work, so basically this is an actin filament, and it's being polymerized in here, so it's being extended continuously. So the idea is that this is converted into motion, so the polymerization ratchets, that's what is normally called, the cell membrane forward. And in order to do so, since you have to push against the membrane, you have to push from something, so you have to be attached. You can't push from a fluid, it's much more difficult. So this attachment or effective friction is provided by protein clusters which are known as focal adhesions. These are very important to understand cell motility on a substrate. And the common view, if you look at, there is a very nice book by Bray on cell motility, or there are very many reviews, is that this motion is triggered by actin polymerization. So there is a problem with this idea if you think about cell motion in a 3D context rather than on a substrate, because in that case, it is not so easy to think of what provides the friction. Okay, so for instance, so this is dubbed cell swimming. So this is another mode of cell motion, although it has been dubbed cell swimming. This is what happens, for instance. So this is a cell moving on a matrix gel. I should tell you that a cell crawling on the substrate does so at about 300 nanometers per second, okay? And whereas a cell swimming, in this case, so these are 4 hours, and this is about 10 microns or so. So it's not moving very far, but still moving. And so this is another example in which the people have taken some bit of a cell from a tissue, put it in vitro, and cell move around here as well. This is a cell motility motion which is relevant in small metastases that they can form in vitro. Yeah, metastatic cells. So the question is, if crawling is driven by actin polymerization, so this sort of rather more exotic way of motion, how can we understand it? How can the cell move in 3D? So again, so this is a couple of movies from Elson which I like, which give the idea. I have already said that to some extent. So if you are moving on a substrate, you are relying on focal adhesion. So these are sort of clusters that have to be dynamic, so they have to disassemble. So if you are moving in this direction, your foot has to detach from here, so they have to disassemble and then reassemble here. Okay, so they have to be dynamic, but they provide the friction with the substrate, so in a way it's like a tank which is moving. Okay, so it needs a lot of friction. Whereas if you knock out the focal adhesion, you would imagine that then the actin, here is only a polymer, but even the network would do the same, would slip, and it wouldn't be able to push forward effectively. A little bit like the guy on the right. So the mechanism of cell swimming when there is not so much friction should be different. So now I will tell you, I will speak 5 minutes or so about a minimal model for to understand this cell motility, if you want. So we'll try to set up a model for a cell fragment. So this is like the keratocyte that we saw before. You can have even simple fragments in which you don't have a nucleus even. And we want to try to understand what's the minimal description in terms of a continuum mean field model, so there will be no fluctuation. And one thing that we have to keep track of is the macroscopic polymerization. So these actin fiber, they actually are polar. They have a plus and a minus end, so they would polymerize here, but we will not worry too much about that right now. Later on, maybe a bit more at the end of the talk. So one thing that you have to keep track is the polymerization so the polarization of actin. Okay, so you have to keep track of their direction. So then, on top of polymerization, you have another important active process in the cell, which is myosin contractility, which is sketched here. So if there are two actin fibers, then a myosin filament can form. So there are several myosin motors which will attach to both ends and contract the network. So to the lowest order, active particles like this need to be force dipole. So the Curie group and Sriram and collaborators and Tani and Cristina have developed a continuum equation, Tani Liverpool and Cristina Marchetti, for this kind of systems. And to the lowest order, since there is no force which is applied to them, this has to be a force dipole. This will be a contractile dipole. Okay, and then by pulling on the fibers, this will create a flow of solvent end fibers that will be dipolar. So if you think about a concentrated solution of active dipoles, you can sum up their forces and find that this is the divergence of a stress tensor. It has to be the divergence because this is a sort of a contact force. Now, so think about a surface. The surface has to depend on the, so the force has to depend on the surface. So it has to be a divergence that you integrate in this, you convert to a surface contribution only. So this stress tensor is minus a phenomenological parameter zeta, which tells you the activity, times P alpha, P beta, where P alpha is the orientation of the actin fibers, okay? And this is the Kronecker delta. So zeta is smaller than 0 for contractile fluids, like we are interested in now. So that's a very important player in this, in the physics that I will discuss about. So activity comes through polymerization, but also through contractility. Okay, so we need then, so in the minimal model, we will be following 3 fields. So one is the polarization of actin, so that tells you the average orientation, okay? Then we want to follow the velocity of actin, okay? So that's V of RT, and then we want also to follow the concentration, okay? Because we want to describe a system in which there is a droplet, so which is a simple model for an actomyosin droplet or a cell fragment. So the system I'm thinking about is very simple. So is a bag of active droplets. So here the concentration is different than 0. Here the concentration is 0. Here we are assuming also that they are polarized. So the polarization vector is different than 0, and here it's 0. And this is all dipoles which are contracting. So in order to, I want to tell you a little bit how to make a droplet which has a non-zero C inside and a C equal to 0 outside. We can do that by introducing free energy. So we can introduce a potential which has two minimum, one at c equal to 0 and one at c non-zero, c0. Okay, then there is another term which penalizes gradient fluctuations, so this will be related to the self-extension. And then I got another term here which changes sign if c is larger than c naught. So if c is larger than c naught, you want to have some p which is different than 0. It's like Landau theory, right? So you like to have positive p, otherwise you like to have 0p, right? So that's very simple. So that's a way to plant a droplet of actomyosin which has a non-zero concentration in its interior. And the other term, K, is an elastic constant. So it penalizes deformation of the director profile, of the polarization. So in order to tell you what we do, I just go through the equation a little bit more quickly now. So this is an equation for the concentration profile. It's been advected, and there is one active parameter which tells you that the actin is being treadmilled, okay? And this is done in the equation by putting this W, P alpha, so this advection by P term. And then the free energy, which wants to make a droplet, will introduce a flux, which is del F over del C, and that will introduce a diffusive term like this. So repeated indices are summed over there. Okay, so that's the equation for the concentration. So you want to form a droplet and you've got flow. Then you've got an equation for the order parameter, which is basically given again by the free energy that I told you before. And then this derivative, since we have flow, will be a material derivative. And the flow field, which is the third field we want to follow, has a Navier-Stokes equation. So this is the MA term, like the inertial term. Then this is the active stress term that I told you about before, which is the main player here. And then you have viscosity. You can also have a thermodynamic stress tensor coming from the physics of liquid crystal. This will not come too much in the physics that I tell you about. So there are two active terms. One is the treadmilling, and one is the active stress. So this equation were written by Luca Giomi, Tani Liverpool, and Cristina Marchetti. Basically this form. Okay, so I now come to the first part of the result. I want to discuss what is the physics of a droplet of actomyosin. And as physicists normally do, I will now switch one term at a time. At first, I will say that there is no treadmilling, okay, because I'm interested in understanding non-conventional modes of cell motility, like swimming. So W equal to 0, and I will put contractility only. Okay? So basically, we have W equal to 0, and we got zeta. And I'm initializing a droplet and seeing what happens. So what happens is that the droplet is polarized along this direction. It first contracts, and it then starts to move. Okay? So it took us quite a while to get to this point. We were quite pleased, because in this case, again, there is no treadmilling, but there is a droplet of actomyosin, which by some other means managed to propel itself. So how does it do that? Well, it does that by exploiting an instability which is already there in the bulk. So what happens is that there are a lot of active contractile dipoles which are pushing the cell in this direction. So it will contract and will become an ellipse. So this simulation, to begin with, 2D. Okay? And then it will splay and start to move. Perhaps I can tell you just briefly why this instability is connected to motion. So if you have a bag of contractile dipoles, or you can forget about the bag to begin with. So as I said at the beginning, we will just contract along this direction. And then suppose that you have a splay fluctuation inside the droplet. So what we'll do, so the fluid is being pushed this way and this way. So the fluid flow is balanced, so there is no motion of the droplet. But if you have a splay fluctuation, then what will happen is that you will have more forces this way than that way. So there will be a force on the system which is pushing to the right. So now if you have a flow which is pushing to the right, this dipoles will be splayed even more. It's like, so I've got more forces here than here, more active forces, and then this dipole will be splayed even more. Okay, so this system will get unstable and will be associated to flow. So that's the basic physics, if you want, that I'm explaining in this cartoon. Yes?
What sets the speed VCM?
VCM, I will come to that. Yes, it's...
Is it steady?
Yeah, it is steady. Yeah. And we'll discuss it a little bit, actually. So another way is this to... Well, we will... OK, let me start. So this is the basic physics which is working here. The total flow is incompressible, that's right. Yeah. So now you will see how it does that. That's exactly what happens. So when the cell moves, so when it becomes motile, it will have a form which is a crescent shape like this, and you form two vortices just because the fluid is incompressible, and they will push the cell forward. Okay, so if you think about the whole of the system, the cell is moving, so the droplet is moving to this direction, and then the fluid is moving in the opposite direction. So the total momentum has to be conserved. So this mode of motility requires, to some extent, coupling with the external flow. And we believe that this might indeed be a possible mode which is exploited by cells when they move in 3D, because there again they can tag the flow which is outside, if it is in a matrix gel, for instance, like here. And this matches the picture that we have on the right, at least qualitatively. So another way to understand this transition, which I thought about more recently, speaking about with Rhoda Hawking, a student in Sheffield, is to think about multipoles. So this is not something we had been thinking about before, but it has, I think, a nice little explanation of the phenomenon. So when the particle is stuck, you got this contractile dipoles which push this way and extend in this direction. So this is so this force distribution, F1, F2, F3, F4, is perfectly symmetric and will not let you flow. This has a dipole, so this is a dipole to the lowest order, so it will be like a giant dipole, which doesn't flow, it shakes, like the microscopic shakers that Sriram Ramaswamy introduced few years ago. So now if I just, you see, if I just rearrange this force dipole, I rotate this F3 and F4, and if I flip the sign of F2, I can have a system which swims. And now the way I've drawn it is a little bit probably what is not happening in reality, because if these are exactly equal, the dipole moment would go to 0 and this would be a quadrupole. But if what happens probably in the simulation is that this is a little bit larger than this, so this would be like a giant puller because it swims from the back. Okay? So I think this is a nice symmetry argument to understand what is happening coupled to the splay instability. Now, coming to the question of the velocity, we can characterize this as a second-order non-equilibrium transition. So if you fix all the other, if you fix basically the elastic constant and all the other parameters, in particular the droplet size, by cranking up the contractile activity, you first are stationary. So this is showing the velocity of the center of mass of the droplet as a function of activity. So first, even if you put little instability, little random noise in your equation of motion, this will splay a little bit and then come back to the state. So it will be linearly stable, even non-linearly actually. Whereas here, you can have, you can form you can spontaneously break the symmetry and splay outwards or inwards and move into different directions. So that's the physics of our motile droplets. So this is known in nonlinear dynamics as a supercritical Hopf bifurcation. So for us physicists, it's like a second order non-equilibrium transition. Okay. So now we can play a little bit with these systems, and the system I showed to you before is contractile dipoles represents actomyosin. So that's the one which is relevant in reality. So if you were thinking about having a extensile droplet on the other hand, this very much works similarly. But now the spontaneous symmetry breaking is a bend rather than a splay. Okay, so the polarization would bend, and then the motion is perpendicular to the bend. Okay, and again, you can understand it with the same, basically, physics. If you flip the sign of the dipoles and you make it bend instead of splay. And also this pretty much works in the same way. The nice thing, so this would be a droplet of pushers. So you would have to have a bag, for instance, of bacteria. So it would be much, much larger length scale. So the cell, of course, is about 10 micron, whereas each of the bacteria will be 1 or 2 microns. So we are thinking about a much larger scale. If you want to think about some way to construct this in the lab, but it could have some interesting properties, because if you now say, so suppose now the polarization, you can see it here, but to the right. So let's introduce moving, so like the treadmilling on top of this extensile activity. So the extensile activity makes you move this way. The treadmilling would make you move along your direction. So here you have a nice interplay. So in the contractile case, they would be in the same direction. Here you see that the self-advection, so the W10 would move you along this direction, and then you can also spontaneously break the symmetry, so the combination gives you spiraling motion or circles. So you see that's a cute curiosity if you want. It's not clear to me whether you can, we can actually do it in the lab, but it's a nice system. And the physics is exactly the same. You just need to play with the 2D geometry. It's quite simple. So now in 3D, we try to do the contractile droplets. So this was important for us because we wanted to propose this as a way to move in 3 dimensions without treadmilling. And you see that it works in pretty much the same way. So this is the shape of the droplet that we would predict, at least with the surface tension we have, should form. An interesting aspect here is, so the physics is pretty much the same, but the geometry now is more complicated. And this is the flow field. So we have just followed here infinitesimal particle of the fluid. So they would just do vortices, but they have toroidal symmetry now. So the interesting thing in 3D that there is a finite range of activity for which the droplet moves. So if you overdo, you can splay so much that you form a hedgehog, and this hedgehog by symmetry is now static, is stuck, cannot move anymore. You would imagine that it could rotate, and indeed it could. So in an extensile case, you would see that, not in a contractile, but it wouldn't go anywhere. So the last thing that I want to tell you about this contractility-induced motility mode is that if you are on a wall, so this would also work. So this is a 2-dimensional simulation in which the droplet now forms only half a structure. So it forms a quite strange bean-shaped structure, and then it starts to move like a semicircle or a hemisphere in 3D. The important thing is that since this is a swimming motion, it has to couple to the fluid. So in reality, most cells experiments are done in a fluid, actually. But one could imagine that the surface tension of the membrane could somehow quench this fluid flow. In any way, so the mere presence of a wall would not hinder completely the motility motion. The problem is that when cells crawl, they do that by sending out the lamellipodium. So it's a very different mechanism. So it's certainly not working in the cell crawling experiment, but it's something that you could engineer again. in the Lamian principle. So I think now I want to discuss the other case in which, for some reason, right, so for some reason it has skipped. So what I want to do now is the actomyosin droplet, in which instead of having contractility, I now switch off contractility for the moment and I have thread milling. So the situation is like this. You got your substrate, and you got a polarized droplet. And now zeta is equal to 0, but W is empty. So this thing can move just by thread milling. And the biology suggests the thread milling only occurs in a very thin layers close to the wall, because this is where the focal adhesion are, and this is where also the proteins which stimulate polymerization are. So we're going to say that W is equal to W0. Okay, so the polymerization is working like this. That's W of Z. So for small thread milling, the droplet just deforms and moves along that direction. And then if you increase the thread milling, even if you don't have any activity, the competition between thread milling on one side, which has this exponential shape, and surface tension, which tries to pull the membrane back, gives rise to a phase transition between a motile but quasi-spherical, quasi-circular droplet and the one with the foot. Okay, so this foot is already quite a lot resemblant of the lamellipodium. And yeah, what I was saying is that at least in the simple model which I showed you before, with the concentration, the polarization, and the velocity field, you do not need activity. So activity is usually deemed necessary in the cell motility paper just to retract the rear. In this case, it's the surface tension which does the job. Okay, so that's a very simple model. Okay. And yeah, if another thing that we looked at, if you plot the velocity of the cell, you can control now the amount of slip which you allow for at the wall. So if there is no slip, this is where thread milling works better. So the case that I showed to you before of contractility mediating movement would be killed by having no slip, right? So if you put no slip, it means that there is a lot of friction, and then you can't put the flow in motion so much. Whereas for thread milling, so polymerization-induced flow, the having no slip makes you go faster. And that is in line with our intuitive cartoon at the beginning, which you need focal adhesion. So if you let the system slip, you lose the velocity. So this is the amount. So 0 would mean no slip, and 1 would mean full slip. So somewhere in between, you lose the motion. Okay, and if you... I'm losing a couple of slides, I don't know why. So, yeah. If you plot the steady state of this contact area as a function of polymerization, you again see that there is a kind of a non-equilibrium transition, and this looks very sharp this time between a protrusion, a state with a protrusion, and a state with no protrusion. Okay, so I will discuss now in the remainder of the talk a few recent simulations, which were done mainly by Adriano, and they discuss the case of 3D. Okay? So the rationale for this is that we have seen that thread milling by itself allows us to understand in a minimal way this lamellipodium, but obviously it's important to understand what happens when you have not a 2D simulation, but a full 3D simulation. In this simulation, you just resolve this dimension and this dimension. You don't know what is happening in the plane, in the direction which is coming out of the board. Okay? So now the free energy that we use to stabilize the droplet is the same. The only thing that I wanted to tell you is that there is another parameter which now plays a role. This beta p dot grad c is an anchoring term which tells you how the polarization of the actin behaves at the wall, and beta larger than 0 will promote anchoring outwards and normal. Okay? That's the one which seems to be suggested if you look at electron micrograph, for instance, the actin network. So keep in mind that now there are 3 parameters which are important: zeta, W, and beta now. So yeah, they come in dimensionless, so you need to think about dimension. If you want to think about dimensionless parameters, zeta is associated to zeta r squared, that would give you a force divided by the elastic constant. So that controls the physics of the first part of the talk, so that the activity number. The relevant number, we're not completely sure, but I think it's eta W0 divided by the surface tension for the second part. On the other hand, this treadmill induced, so this is the capillary number. And beta is the normal anchoring, so this is the first parameter, the second. And beta gives you an anchoring W, which then gives you a dimensionless anchoring that we are all familiar from liquid crystals. So these are the 3 parameters. Okay. So let's see what happens. So if beta is very large, then what happens is that the polarization forms an aster, and the droplet forms this fried egg shape, which is actually found in many cells. So for instance, static keratocyte would be very similar, where the droplet is static and doesn't know where to go. So you form your lamellipodium in all directions, if you want. Now, if, yeah, keeps on. So if the treadmilling is dominant, so if your anchoring is small and the activity doesn't play much role here, you form a structure which is this from the top. So this is the one with the foot previously. Yeah. So you see that it looks good in the sense that we are comparing with a cell moving with a lamellipodium, so the lamellipodium would fan out. So it looks good on the side, but it looks a bit funny on the top. And if you look at the dynamics, this is what happens. So you form a nose, but this is not very much like a lamellipodium. So if you increase the anchoring on their ways, and so when all contributions are important, then you can form a droplet which is very much similar to the moving keratocyte. So that's nice because there are no simulation previously I know of full 3D motion, which is similar to that of a lamellipodium. And to get this, you need to switch off all your contributions. Switch on all your contributions. And there is quite a lot more, actually, that we are discovering. Every couple of weeks, we discover another state. So this is another one which we have for intermediate value of the parameters. So there can be oscillations. So this is, you form first the structure with a nose, and then you form this other bifurcating structure, and then it goes back and forth between these 2. Okay, so you got oscillatory dynamics. So this ends what I will just recap what I say, and either we have questions or I have another few movies of a related problem. So the first part of the talk discussed a new way to provide motility to an actomyosin droplet, which I hope to have convinced you may be relevant. Though it's a simple model, very relevant for cell swimming. This is a cell which was swimming in the matrix gel. And the way by which this happens is that you splay your actin fiber and you create flow spontaneously inside this droplet. So if you use treadmilling, which is localized at the surface, okay, so you've got this shape here, On the other hand, you have another non-equilibrium transition between a droplet which moves without any protrusion and a droplet which moves with a protrusion. And if you include both activity, treadmilling and also this anchoring term, which tells you how the fibers behave close to the surface, you can reproduce shape of a crawling keratocyte quite well. I think people had not been able to have a model of a 3D droplet which resembles a lamellipodium. So that could be used to work on crawling cells, I think. Okay, so I think that's it for me. And as I said, 5 minutes or so. Yeah, we can discuss. Or I have a few more. Still power.
Hi, thank you. That was very beautiful. Now, I have two questions, actually. The first one is, if you have like an argument to explain this pancake type of structure that you form in the lamellipodium when you switch on anchoring. And the second one is that how does the velocity of the cell scales with the size of the cell?
Yeah, because I forgot to say that. Yeah, so okay, that's good. So the argument that I have is only that if you form this lamellipodium, and then you have the anchoring at the thin two-dimensional sheet will be happier if you make it larger. So if you, instead of having something like this, you have something like this, the anchoring will be happier. Okay? So that's the only argument I have.
Can you raise the board slightly?
Yeah. It's not very profound. So instead of being like this, it's have a lot more surface. Yeah. So that's the. I could imagine that this will stabilize the lamellipodium, and the flow, the activity will lead to further splay actually, which will in a way have a feedback on this, and surface tension will try to shrink it. So beta and activity will try to fan out, and surface tension will try to shrink, I think. The second question is interesting. So the, yeah, as you said, as you saw, well, there is a transition, an equilibrium transition, so it depends where you go. So, I mean, I can answer for this bit. Okay, so the velocity of the center of mass, but if you think dimensionally, so one of the four, so the force which is in this droplet should scale like zeta r square. So zeta, but sometimes people, so this is a stress anyway, so that will be Pascal. So if you Newton per meter square, so if you measure for the psi square, if you multiply by psi square, you get a force. So this will be the typical force that is pushing you. It is probably going to be like psi r square, or I'm not sure. It could be something like this, right? But at the large zeta will be psi, zeta square. And then if you want a velocity, it seems reasonable to divide by an effective viscosity times r, so it should scale like the size. And that's more or less working. But you have to go very far from the transition to lose the bifurcation behavior. Yeah.
Just a question about the treadmilling.
Yes.
So you've assumed that the flow of monomers required to do the treadmill is restricted in this. Well, in the 2D case, you essentially had it right at the surface.
Yeah, that's right.
But in principle, one should determine that, and there'll be a flow field inside, which depends on various things. I mean, it's not obvious that the monomers are all drawn from right near the surface, and because of viscosity and whatnot, it should be spread out in some region.
Well, I mean, if you try to polymerize as soon, so this is something which is known as ruffle. So if you try to polymerize and you are not stuck, it goes this way. So so from the experiment, it's very clear that there are some cases in which the fibers which are not polymerizing are not stuck, and then they do extend, but they are very quickly retracted. So they do need something on which to.
That's not what I mean. Is you assume that the surface is actually advecting at the surface.
You're assuming that the droplet is being advected.
But it's not being advected at the surface.
Well, you have
What's,
the...
what, let me,
yeah, yeah.
what's being advected is monomers inside the droplet
Yeah.
come around to the top. And so you assume that that advection of monomers is occurring inside the cell. It seems that you're assuming all of that advection is
Yeah.
occurring at the surface so that you get effectively a slip boundary condition. But what I'm saying is it's not obvious that they should be advected along the surface.
Yes, okay, so in order to do a better model, you would have to then, so this is a very, if you want, practical way to put in the biochemical information which we have, because the actin nucleators will not be, so they are not going to be here. So the actin nucleators will not be here, they're not going to be there, you know, so that's, if you want, I agree that you have to put more knowledge into the model.
I mean, I would have thought you put a sink on the right, a sink on the right, and a source on the left.
Yeah, but you have to also put it on the surface. Otherwise, it wouldn't work. Yeah, we tried various
Maybe
possibilities at the beginning.
maybe we can take a few more questions. Please, very short, both question and answer. I have 3 here.
A very lovely talk. Enjoyed the simulations. So the steady state structures that you're observing, you have in some way a fast time scale, the time scale of relaxation into of the concentration in the order parameter P into the steady state, so to speak,
Yeah.
and then a slow dynamic. So I'm wondering whether you've thought about the singular limit problem, where you basically have just a free boundary, the interface, and the effective law of motion for that interface. It would seem to be a driven version of motion by mean curvature with additional structure.
Yeah, no, we certainly haven't, well, I haven't thought about this. It's true that there is this separation, so you have to, so that's a good point to pick up on, I guess. We should have a chat, yeah. We didn't think about that, yeah.
Would you expect any qualitative differences if you would include like bending of the membrane?
Bending of the membrane, yeah, it's possible certainly to add it. Not for the first part of the talk. So there the membrane is very much a passive player. For the second part, it's not clear what you, again, in the 3D it might have an effect. And we know how to put it in, it's just that it's another parameter, but yeah. So there's something, if you want, yeah, this is only a stretching, rather. So this will only have a surface tension, this membrane. You can have a Laplacian C squared, and that's an interesting thing to add, yeah.
One final question?
Well, very short. Exactly this bifurcation diagram.
Yeah.
What breaks the symmetry left-right? Because
Noise.
cell decides. Is there some additional mechanism? Which
No, well, but it's just a standard, like in the IC model. You put some noise, and sometimes it goes, plays the outward, and sometimes it plays inward. So there's just noise, at least in the simulation. In reality, I don't know. Yeah, in the simulation, there is only noise.
Very well. You can thank Davide.